I guess we should probably get started and hope you guys enjoy the meeting. My name is Charlene Zhang again. I would like to thank the organizer for giving me the opportunity to introduce today's keynote speaker, Dr. Lingling Chen. So this is really a great honor. I think this is the best job I can expect as a session chair, I would say. So Lingling is an old friend and a colleague of mine. I admire very much. We know for already quite a few years, actually, from this very meeting when we were both trainees. So it's, if I remember correctly as well, Jim Watson actually said that people here at Cold Spring Harbor, they go by first name. And he mentioned that he did not like some people at Columbia who were sued and go by last name. I'm not sure whether the latter is true, but anyway, I will go by first name. So Lingling finished her undergrad in biology and a master's degree in pharmacology in China. And then like many of us, she came to U.S. for graduate studies. She performed her PhD with Gordon Carmichael at UConn Health. So she finished in 2009. And at the same time, she also got an MBA. So trainees, think about it. If you want to be super successful, this can be very helpful. Yeah, so she then stayed in Gordon's lab briefly as a postdoc, but soon was promoted as an assistant professor. So that was 2010. And in 2011, she decided to go back to China and join Shanghai Institute of Biochemistry and Cell Biology, which is part of Chinese Academy of Science, to launch her independent career. She's currently associate director of the state key laboratory of molecular biology since 2017, and now the founding director of the newly established key laboratory of RNA science and engineering, or RISE. So Lingling's research has primarily focused on biogenesis and function of different forms of long non-coding RNA, and really is a pioneer in this field. But her PhD work really started from studies of protein coding mRNA, but with inverted ALU in the 3' UTR. So her work really showed very nicely the inverted ALUs were sensed by double-strand ion binding proteins, and with interesting interplay with long non-coding RNA, NIT1, and that regulate expression of and export of those mRNA and expression and translation of these mRNA. And so the theme is, RNA structure, long non-coding RNA, gene expression, function, and you can see this will come up again later in her research. So that was the time that RNA-seq was a revolutionary tool to study gene expression, unlike many others who focus on polyadenylated RNA. And Lingling really tried to, also examine those without polytail. And she noticed that, for many of the genes, actually there's a lot of RNA-seq reads in part of the intron, and for some of them, and very interestingly, the SNO RNA sits right at the boundary where the RNA-seq reads accumulate. So this led to the discovery of SNO link RNA, which is very interesting, and those link RNA are protected because of RNA secondary structures at the very end. And then, that was the time circular RNA was discovered by Julia Salzman through back splicing, but the mechanism was unclear, and the Lingling's work really showed very elegantly like the inverted ALU at the boundaries near the splice site will help to bring them together to facilitate circularization of RNA. So this is really, really nice work. But importantly, her work goes beyond biogenesis, but also show the physiological functions of those circular RNA and link RNA, such as the circular RNAs can fold in specific ways that can be sensed by the cell through the double-strand RNA sensing pathways, which play an important role in immunity, which can be disrupted in autoimmune disease, and SNO-link RNAs can play an important role in sorting and processing of the ribosomal RNAs and organization of nucleolus. Of course, I'm not going into detail. And so she published so many important papers, and she has been in the editorial board of many journals, including Science and Cell. Actually, at the very beginning, when she published important papers, I would like to send her a message, say, congratulations, but then I stopped. Why? I realized I cannot keep up with the pace they publish the important papers. So, surprisingly, she won many important awards. I'm not going to tell you all of them, of course. I'm just list a few. She was the HGMI International Research Scholar since 2017. 2021, she won the Mid-Career Award by our own RNA Society. And then very recently this year, she was one of the new cornerstone investigator, which is the Chinese version of HGMI. So without further ado, I'd like to congratulate Yinglin for all the amazing work again, and look forward to the seminar.
Wow. Thank you, Charlie. That has reminded me my first talk in the meeting. That was 2009. I talked about my graduate work on NIT1 RNA. And over the last 2 days, we actually heard many talks about the graduate students. So I want to say thank you to the organizers for giving me this honor to deliver a long talk. So my lab works on link RNAs and circular RNAs. But if we look at the abiogenesis, at least for the molecules we work on, actually came from eukaryotic mRNA processing. So thank you for giving me this opportunity to talk about these molecules. So back to a decade ago, the application of RNA sequencing technology has, I'm not sure, it doesn't work. Okay, here we go. That unveiled the pervasive transcription of the genome, yielding over 10,000 non-coding RNAs. Those RNAs are mRNA-like, except NIT1 and MALA1 at the time. So Jeremy Willis, he was a graduate student with Derek Spector at Cold Spring Harbor, and he uncovered that the three' ends of these two RNAs are stabilized by triple helix structure. And when I was Gordon, I worked with this link RNA as well. So these serendipities have allowed me to have the hypothesis and ask questions. Maybe there are some RNAs don't have polytails, therefore have been made from the polyadenylated RNA sequencing that was enriched by OliDT selection. So as soon as I received the very first funding from the state of Connecticut, we began to develop this non-poly RNA sequencing. So I wanted to give a special thanks to Gordon and Brent, my computational collaborator, Li Yang, at that time was postdoc with Brent. So both of them are very generous. They allow us to explore new directions. Looking into the non-poly RNA sequencing, we found many signals from introns and exons, and we call them excised introns and excised exons. So the question was what mechanism can keep these RNAs from degradation? So while we started the project here, and then I had my lab relocated to Shanghai, in my lab we found if some intron contains two SNO RNAs, that during splicing, the formation of SNO RNA protein complex at each end can protect the intronic RNA from degradation by exonuclease after debranching. While some other intron, if they have the C-rich motif, okay, close to the black branch point site, that can have this very inefficient debranching, leading to the formation of second intronic RNAs from layered introns. And meanwhile, actually also look at non-poly sequencing and also develop the RNA-enriched sequencing, have identified the widespread expression of the circular RNA from backspliced exons. And we also worked on these RNAs. Together with Jeremy Willis' group, we have confirmed that in order to facilitate the distal RNA backsplicing that often require the appearance of a pair of complementary sequences. And then later on, by looking for additional SNO link RNAs, we unexpectedly identified SPAR, which is 5' SNO RNA and 3' polydenylated processing from polycistronic transcripts that coupling with very fast port2 transcription and a weak upstream poly signal that ensures this fast port2 can first reach the SNO RNA to co-transcriptionally form SNO RNP complex, prevent this XRN2 trimming. So all of these RNAs are seamlessly connected with eukaryotic MR processing. Now the question is, what do they do? So over the last 10 years, we've been focusing on the nuclear retained RNA because they don't have the cap or tail. So once they are made, they actually look like the nucleus. We also worked with circular RNAs. I want to use one example to show you how we're chasing the link RNA function by developing the cell biology-based approaches. So here, we all know nucleolars. This is the nucleolars, which is the biggest nuclear condensance. So that has 3 different sublayers: fibrillar center, disfibrillar components, that constitutes the fundamental unit for ribosome DNA transcription and ribosome RNA processing, and then assemble into ribosome in the granular components. So over the years, now we're able to use imaging to capture these FCDFT units. The same staining marker, fibrillarin, in the DFC region. This is textbook. So now at this resolution, we're able to dissect this long-non-coding RNA called SLERT that can enhance pro-ribosomal transcription at each FCDFC level. What we have shown that with this SLERT RNA, that is required to maintain the correct size and liquidity of each individual FCDFC unit. However, without this SLERT RNA, that can lead to collapse of the FCDFC units. Just wanted to remind you that rDNA, this active rDNA, transcription occurs between the fibrillar center and disfibrillar center. So we took the single molecule turf assay. For example, this rDNA on the surface, we put the DDX21, which is interacting protein of the SLERT RNA. We observed striking hijacking effects of this protein complex to this rDNA, while addition of only 1% of this long-non-coding RNA that can successfully prevent this DDX21 in clusters that hijacking the RNA. So that perfect explains how this RNA works. That on the one hand, that can keep the right size of these FCDF units. On the other hand, can prevent DDX21 hijacking transcribing RNA. So this type of assay we can do with nuclear retained RNAs. And at this resolution, we recently have uncovered another layer in the nucleolars, which we call them peripheral DFC region. So this region enriches 12 proteins, 12 nucleolar proteins. As we know, nucleolars is the most prominent nuclear condensance, but all these 12 proteins, only 10 of them have mobility. 2 of them are static protein, including this URB1 protein. So this protein is required for the integrity of PDFC. It's very large, more than 2,000 nucleotide amino acids. So as a PDFC, URB1 can provide a platform, allow the 3' end pro-ribosomal RNA to attach and form correct conformation, and then allowing the U8 SNO RNA to recognize and subsequently remove this 3' end of ribosomal RNA. That is essential for life. So this is what we've been doing for this nuclear retained RNA. We also work with other SNO link RNA, which are absent in the patients with Prader-Willi syndrome. But I think maybe because we've been developing different assays, we want to look at the RNAs better. So here are several assays that we've been doing. So this is just for people who may be interested, want to use the RNA imaging to look at your RNA. This is DK13 system. On the 1 GRN allow us to do the tracking of the NIT1 for paraspecific fusion. And this is the in vivo tracking of RNA in fish embryo. Excuse me. I just wanted to show you one more movement. So this allows us to do the de novo transcription of the endogenous. So this is very much heterogeneity. But here, the second cycle become more homogeneous. So this is the transcription memory, and also allow us to say mRNP export, and we can also label the RNA. But this is only restricted to abundant RNA for 3 colors at the same time. So all the plasmids have been deposited at G. Well, my talk today is actually going to focus on circular RNA. So apparently, circular RNA are so different from long RNA. So how can we study them? Well, I want to first give you some brief introduction for those who are not familiar with circular RNAs. So this is the very first circular RNA captured by electron micrograph. That is the circular variant RNA from the plants, circular or the tiny racket shape. And then in the 1980s, many circular intermediate RNA were found that can be processed from non-coding sequences, including the introns of Archaea, ribosomal precursors, or Archaea tRNA precursors. Now, the autocatalytic introns have been well adapted for initial synthesis of circular RNAs. So this is actually a review article recently Lee and Jeremy and I put together for the annual review. And also, we call this circular RNA processed from RNA POL2 transcribed pre-RNA as a spliceosome-dependent manner. So introns and exon-produced RNA. So now, this is the first circular RNA of this type captured in HeLa cytoplasm. And here shows in the early 1990s, a handful of those RNAs were reported, but they were thought the byproducts of the spliceosome. So now we know they're widely expressed. And among these two different and other type of circular RNAs, those ones came from exon back splicing represent the most prominent class. So I'm going to focus on this class today. So efforts from many labs have shown that this type of circular RNAs are widely spread expressed, but generally at very low levels and exhibit cell and tissue-specific patterns. And they are differentially expressed during development, immune responses, neuronal differentiation, and some disease contexts. For their biogenesis, that requires spliceosome machinery and is modulated by these cis complementary sequences that can bring distal splicing sites to close proximity to facilitate back splicing. And meanwhile, can also be regulated by trans factors. So they are very stable once produced, especially for those without the introns. They are largely localized in the cytoplasm. So many of them have been annotated as non-coding RNAs, but recent study has also shown that for those containing either internal ribosome entry set or M6A modification, I believe Zefeng is going to give a talk tomorrow, so that can encode to protein. All right, so although this is often flanking with the inverted allo repeats, but it's not sufficient. It really depends where this repeats is forming. For example, if the repeat or the pairing occurs within individual intron, that still leads to canonical splicing. But if the pairing occurs across the future circularizable exons, now they can form the circular RNAs. We had demonstrated this idea using expression assays. So let's focus 2 and 3. So these two circular RNAs can be circularizable in the future, flanking by one pair of fully complementary sequence. We put this cassette in a split EGLP, which was a gift from Zefeng. He made this very useful plasmid, I think, when he was Chris. And then we see in this cassette the strong induction of circular RNA, introducing competition over here that can diminish circular RNA formation. So because of this regulation, I wanted to say this is the cassette I'm going to use for in vivo in mouse model in my later talk. So because of this wide-distributed allos and the competition of pairing in human introns, we have observed the phenomenon called alternative circularization, that multiple circular RNAs can be produced from the single gene. Here shows the very famous gene called DMP3B. Computationally speaking, more than 10 circular RNAs can be annotated over here, and for those highly expressed ones can be detected by northern blotting. So and later on, together with Lee's lab, we have characterized the different 6 types of alternative splicing, including alternative back splicing events, 2 of them, and all the 4 types of classical alternative splicing events can be found with circular RNAs that contain multiple exons, including intron retention, cassette exon, alternative 3' and 5' splice site selection. And all of these RNAs, they have the same back splicing sites. So very recently, we call attention to the field that for people who work with individual circular function, so please also include the exact exon to your first circular RNA. So for example, this came from exon 2 and 4, this is from 2, 3, 4, and some of them may have the retained intron, and some of them may have critical splicing site to resolve any potential confusion by people who work in the different circular RNAs from the same gene. So big question is, what do they do, right? Because they have sequence overlapping with linear RNA, so make it very challenging to study individual circular function. So we've been trying to advance methods to study them all. In the same time, we wanted to look at them as a whole. What is the key features do they have? So one of the questions we asked in the very beginning, so if the cells already produce linear RNA, why do they still to keep these circular RNAs with the same sequences? So we captured the nascent RNAs that allow us to compare the kinetics between splicing and also back splicing at the same gene lockers. We found compared to back splicing, compared to splicing, back splicing events is only less than 1% of that of canonical splicing. So it's very slowly produced. But some of the circular RNAs are very stable, can accumulate to high levels. In addition, we use chemical probing to detect the circular RNA within 500 nucleotides long, because on average, if you look at all the circular RNA produced in the cell, the average length of circular RNA is about 500 nucleotides long. And we found many of them can contain this intramolecular short upstream RNA region that allows them as a group to interact with the protein called PCAR, which I'm going to show you in a minute. Although they are stable, but they can be rapidly degraded when the cells infected by, for example, poly-IC to mimic viral infection or treated with the virus. But the endonuclease called RNSL, which is active during this process, and upon this treatment, the half-life of circular RNA is only about 1 hour. So because of this biogenesis, we actually proposed a model that in normal situation, the different circular RNA, if they have this short upstream region, they can bind to the protein PCAR to keep them as an inactive state, while upon infection, the global circular RNA degradation can release PCAR to be involved in antiviral activity. Importantly, the misregulation of this process is found in patients with autoimmune disease, for example, lupus, which is the prototype of autoimmune disease. So here we see the global reduction of circular expression, the very subtle RNSL activation, and active PCAR. So what is PCAR? PCAR is a key factor of the host innate immunity. So upon cells infected by the viral upstream RNA that trigger dimerization of RNSL, the active RNSL can degrade viral and cellular mRNA and also circRNA. And the intermediates of these different type of RNAs can trigger those immune sensing receptors leading to activation of interferon. In the meanwhile, the viral upstream RNA can also trigger a subset of nucleic acid receptor with direct antiviral activity, including PCAR, which normally is monomeric status, have 2 upstream RNA binding motifs, one kinase domain. So upon binding to the long upstream RNA, then can form the dimer and to become autophosphorylation. The short upstream RNA within 16 to 33 base pair can block PCAR activation, including the circular RNA I have just shown you that contains the short upstream RNA region. So importantly, now we collect the peripheral blood mononuclear cells and T cells derived from the lupus patients and treat it with the vector I just shown you before to the cell. We see dramatic reduction of PCAR activation and its downstream cascade, including interferon beta trace gene expression. And those genes have been used as diagnostic marker of the lupus. So 4 years ago, we were very happy, to see this result because it suggests somehow a potential of using circular RNA as therapeutic strategy in pathological context with excessive PCAR activation. The big question is, can we do that? So I'm going to talk about basically 2 questions. Those are unpublished work. So first of all, can we make the RNA circles to dampen PCAR activation? And also, can we find the in vivo context that allow us to do the proof of concept? So I would like to thank a group of people, Guo Sikun, Liu Chuxiao, Xu Yifeng, Wang Xiao, and Nan Fang. It's really a group of work because we've been trying so many different techniques that we've never been done before. So first of all, if we want to make the circles, they have to contain the short upstream RNA region, have to have little homogeneity without modification, and also the better be very stable. And also we want to do the animal model experiment, we want to have the RNA made in high yield. We have to have experiment to show the PCAR and the circular RNA have robust interaction and suppression. And also we have to find the correct pathological models, and if possible, we want to do the organ-specific delivery. So, cut this long story short, this is published last year at Mao Cell. So we actually tried to synthesize circRNA aptamers, also the translatable circRNA with the internal ribosome entry sites with different ways. We can do by T4 ligase or by different promoted group 1 intron. Those were published by different labs. But what we have found is different circRNAs synthesized by different strategies have different conformation, immunogenicity, and yield. So, for example, the T4 ligase-based circular RNA that can retain the step 3 RNA region as desired. And these RNAs, they are very good PCAR inhibitors, have more than 1,000-fold higher suppression than PCAR small molecules. Why? That is because the current existing small molecules of PCAR that targets the kinase domain. So that's not very robust or specific. And this type of circles also are very stable in cells. However, if we put this permitted group 1 intron IC, often can introduce extraneous fragments that can distort the final RNA circles and also trigger immune responses. And therefore, they are less stable than the ones that did not produce immunogenicity. But the good thing is, so the catalytic intron can produce the circRNA with high yield. So we wanted to basically balance the circRNA folding immunogenicity and the yield by the autocatalytic group 1 intron. And we have different trials, and here shows one that actually, number 5, that shows much higher suppression on PCAR induced by upstream RNA that's made by the group 1 intron. We call it efficient PCAR inhibitor disc RNA, APIC. So why this is so good on the PCAR suppression? So for years, we wanted to do circRNA and PCAR structure analysis, but unfortunately, the RNA is still too large for correct structure, and PCAR and circRNA complex is too small for correct EM, so we have to turn to a different approach to work on that dynamic interaction. So here is a single molecule turf assay. We have different types of RNAs. The linear upstream RNA, 79 base pair long, or 33 base pair long, or the disc RNA, or the circRNA without upstream region. So they were labeled with biotin and also Cy5, and then immobilized on the turf surface of the imaging. And then we put this Cy3 labeled PCAR. So we were able to visualize numerous co-localization of the protein and the RNAs, but the binding patterns are strikingly different. So if you look at the long upstream RNAs, basically we can see the on and off pattern with about 20 seconds as the stature, as one peak. For the short ones, about 10 seconds, but those are transient binding events. However, if we put this disc RNA with PCAR, in the 120 second of examine window, we didn't see any off of the PCAR. So it's very stable binding events. However, for circRNA without upstream region, it does not bind to PCAR at all. So next, we put this washing buffer to prevent additional PCAR binding that yields the survival probability assay. So the clearly suggests how strong and robust of the disc RNA bind to PCAR. So this data suggests that the disc RNA can form certain conformation to keep PCAR from deassociation upon binding, allowing them to robustly inhibit PCAR, which I think is very interesting for future development of circRNA-based aptamer to target PCAR. So now, how about in vivo? Because previously we only do the cell-based assays. So we turn to cirrhosis. Cirrhosis is a common chronic inflammatory skin disease. It has both autoimmune and autoinflammatory features. And more importantly, as the lab focused on cell biology and molecular biology, and this model is easy for us to handle because we can treat the mice on the shaved back with the drug called imiquimod, which is actually an agonist towards the toll-like receptors. So we treat the drug for 5 consecutive days, and then we're able to see this epidermal thickness that can mimic this cirrhosis. And this model has been well used in the cirrhosis field. So the question was, is this RNA circRNA PCAR actually dysregulated? So we collected total RNAs and protein from spleens of this of the MQ mice, and we found this transient activation of endonuclease RNSL, it has to be very transient activation, as we have observed here. Otherwise, it's going to be a disaster for the animal. We observed global circular reduction and the starting of PCAR activation the second day upon this activation of RNSL. So it's surprised to us that nobody ever really looked at the gene expression wave in this model. So we took advantage of our sequencing data to look at this expression model. So we found PCAR represents one of the factors as a group of fast responding genes, including not only PCAR, but also REG-I-like and NOD-like and TOL-like receptors. The second wave of gene expression, class 2 and class 3, they actually last longer, that are enriched in pathways related to translation and metabolism, suggesting the changes of cellular homeostasis after this very rapid innate immune response triggered by the class 1 genes. And class 4 genes are enriched with auto-inflammation-related genes consistent with the cirrhosis model, and class 5 and class 6 genes enriched in pathways related to cell cycle DNA replication, just a long-term thing. So importantly, we generated the PCAR knockout mice and treated the mice with the same way. In the mice without PCAR, we see dramatic reduction of these fast-responding genes in this immokumab-treated mice. So suggesting PCAR is a crucial but a previously overlooked axis in cirrhosis pathogenesis. In addition, and also importantly, we found the same activity cascade in lesional samples of cirrhosis patients, suggesting the conservation of this pathway. So PCAR involved here. So how about circular RNA? If we in the future wanted to do any circular RNA related with the disease, are these circular RNAs tolerable for the animals? So we generated the first circular knocking animal. So this, again, the cassette I mentioned to you, we put it in the famous ROSA 26, the classical knocking lockers, driven by two different promoters, CG promoter and EFF alpha promoter. We actually see the pan-circularization in different tissues by northern blotting. Actually compare the circular RNA formation and linear RNA. It shows even higher circularization efficiency. That suggests without this intronic parent competition that can induce a strong circular RNA formation. So, because long story short, we have examined different phenomena, related to the animals. And basically, we have seen that mice with disc RNA overexpression appear normal during development. So that's good for the future development of any circular-based drugs. So we treat this disc RNA the same way as we did the wild type, and we found supplement with this disc RNA led to reduction of the first PCAR and the reduced expression of the fast responding inflammatory factors. So now we have the genetic data to support the PCAR and disc RNA in vivo. So can we do spleen-specific delivery? So in collaboration with Dan Pierre at the Tel Aviv University, we're able to do the IV injection of the mouse tail to largely deliver to the spleen of the circles, not to liver, not to lung, not to kidney, but largely in the spleen. And in spleen, northern blot shows that compared to the linear RNA, it has a longer half-life. And the good thing is it has longer half-life, but it still can be degraded in the future. So 48 hours later, only about 5% left. Because we wanted to use them as aptamer, we really care about their folding conformation. So we did the circular RNA shape map again to compare the circular in lipid nanoparticle or not. So they still keep this imperfect upstream region. So now we put this RNP encapsulated RNA 1.5 day of this immokumab model, and next day we sacrifice the animal and analyze the epidermal thickness. And we look at this thickness, we found at 50 pmol concentration that can lead to about 40% of reduction, of the epidermal thickness. So we're excited to see this in vivo data, and also similar PCAR phosphorylation reduction was also observed in PBMCs isolated from the patient. So this in vivo data suggests that circular RNAs with short upstream RNA structures are robust PCAR inhibitors that can dampen excessive PCAR activation in vivo. So how about other model, more dimensional model, for example, Alzheimer's disease? So by looking literature, we're surprised to find that there's lots of literatures have shown the PCAR and phosphorylation of PCAR accumulation in the brains of FAD mice and also in the AD patients. So I don't need to introduce more about the Alzheimer's disease, but I wanted to do a little bit of introduction of the FAD mice. So this is mice normally used or frequently used for people who study Alzheimer's disease. These transgenic mice overexpress the mutant human amyloid beta precursor protein, APP, and also with other 4 human genes carry the mutations related to Alzheimer's disease. Therefore, this FAD mice show accelerated AD progress, including intraneuronal aggregated beta amyloid at 1.5 months of age, and also shows this neuronal loss and also deficits in spatial learning starting at 6 months of age, followed by severe cognitive dysfunction at 7 to 8 months of age, and then neuron degeneration at 10 months of age. So I want to say this project started a long time ago. So she was my graduate student. Now she's my postdoc. I want to finish this project. So anything with the aging animal takes a long time. And Bowen's postdoc in the lab, so she works with the behavior assay, and Sinan work on the computation analysis. So in our hands, we basically collected the mouse individual ones, wild type of FAD mice, from 1 month all the way to 10 months. We see this progressively increase of PCAR accumulation and PCAR phosphorylation in the hippocampus tissue of the mice. So we did a cross. We generated this FAD knocked out with PCAR. We see dramatically reduce the inflammatory gene expression and reduce the activation of microglia. And we know microglia are the primary players in the neural inflammation. So this green color means beta amyloid plaques, and this red color IBA1 is a marker of microglia. And then how about the behavior? So this is a typical Morris Water maze assay. So we put the mouse over here, and we do the 5 days learning process. So for the wild type one, has a good memory and spend less time reach to this platform at the 5 day of training, while FAD shows drawback over here, knock out the PCAR that can rescue this learning process. And on day 6, we remove this platform and then calculate the percent time spent in the quadrants. And again, without the PCAR, that can enhance its time spent in this quadrant. And also, as a control, all the mice show very similar swimming speed. So this actually has been in the same or consistent with a publication showing 2019 that PCAR knockout of the same model reveals beneficial effects on spatial memory and brain lesions. So now we have this model. So how about the small molecule? So we treated with small molecule, target this kinase. With different doses, we see these mouse, they have the clonic convulsion, followed by some animals died afterwards. So suggesting it's not going to work. So how about disc RNA? So to do the disc RNA now, we basically turn to the adenovirus. So we wanted to do different approaches. One is hippocampus steroid tactic injection with one type of AAV. We also do this IV injection with another type of AAV. So we do 2 different siRNA with the structure. One is the one talked about, another one called CAMSEP1. So this shows a northern blot that we're able to overexpress this human-specific siRNA in the brain, and there are about 200 to 500 copies per hippocampus neurons after our quantification by comparing to our cells also overexpress the human siRNA as a reference. So here is the result. So basically, this is a whole hippocampus. The red color shows the beta amyloid. So this is the wild type. This is the FAD mice. This is supplemented with the 2 different types of disc RNA that can reduce this beta amyloid accumulation. Similarly, if we do this neuroinflammation, it's the same thing. This is the hippocampus. Wild type, almost no microglial, lots of microglial, and with 2 different siRNA. Now we see reduced microglial activation. And in addition to this image analysis, we have also looked at this we collect the RNAs from the microglials of different conditions. This wild type, this is the FAD mice, shows upregulated inflammatory gene, and supplement with this disc RNA that can rescue this upregulation. And finally, behavior, that's the key, right? So let's only focus on 2 lines, so that can largely rescue this training process, the blue color and the purple color. This is purple trial, the same thing. I'm going to show you this movie. So we put this mice in this equipment. So here is what type, this mouse, remember clearly it was something before. And here, this FAD just wandering around this entire equipment. And here with the FAD, that supplement with the disc RNA that knows much better compared to the FAD. So that can increase the spatial learning capability of the mice. All right, so I think they're very useful. They'll probably in the future can do something with the disc RNA. But question remains, so how about the other AD models? And can we do LNP encapsulated upstream RNA delivery, for example, by intrathecal injection? And can we make a better disc RNA with a smaller size and even more robust inhibition on PKR? So I think this is how we work with the RNA. One function that is PKR for 10 years. So we know this linking CKR process and function allow us to find they are slowly produced and fast degraded. They are very stable, have unique conformation, allows them to dampen PKR-related inflammation. And we're able to synthesize CKR lack of genicity. And they probably represent a new type of RNP in certain pathologic contexts with unwanted PKR activation, showing here cirrhosis and Alzheimer's disease. So it's about 10 years of CKR study, right, from rediscovery to biogenesis and functional implications. And over the 10 years, many efforts have been taken by so many labs in the field. We have developed a new approach to better discriminate CKR from the linear RNA. We found the new functions. I only talk about this one PKR function to you, but we and others have shown that the CKR can actually also somehow resolve this alpha region to promote POL2 transcription or POL2 elongation, or to promote the exon skipping. For certain very abundant CKRNA, they can work as protein decoys and microRNA decoys. A few of them can work in the organelles, such as mitochondria. And also, the CKRNA can outcompete with the linear RNA for functions. So finally, with the evolving new technologies, we're able to realize CRISPR-Cas13D-based assay to discriminate CKRNA from linear RNA. That allows us to identify this oncogenic CKRNA, and also in collaboration with my colleague Li Jinsong, so we're able to find the first CKRNA that actually play important roles in early mouse development. So I think CKRNA probably can be very useful in the future, especially with a subset of CKRNA are translatable, so we can do lots of engineering with the CKRNA. So because they are stable, they have this unique conformation. They can be used for biomarkers and aptamers, and also maybe substitute or in supplement with mRNA-based protein expression platform. So with this, I like to, my time is perfect. I think almost, I was so nervous. So this is my lab. I select my people in the lab. This is the Bang area in the Huangpu River. This is my institute. My lab is located over here. I like to thank all the collaborators from the computational analysis, the mouse biology, ThinkMark turf, patient samples, image acquisition, funding agencies. Now, in my institute, as Charlie introduced, we have faculty position open for RNA biology. So thank you all. I'm open for questions.
Thanks for an awesome talk. So based on sort of your last slide about the new functions, I imagine that during a viral infection, right, you showed that it induces RNA cell activation so that the double-stranded RNA can be sensed to decrease that double circRNA level, right, and then the double-stranded RNA sensed inducing PKR. I'm curious whether in your psoriasis models or in your SLE models where you show the dampening of PKR, you've gone back and on top of that infected these mice with viral double-stranded RNA infection to see if they can outcompete? Because early on you showed that strong double circular RNA preference.
Yeah, actually, this is a good question. So in the cell model, I can show one slide because this is the common thing for any treatment that basically regulates of the immune responsive pathway. So here is anti-IL-17 therapy, all right? So for patients with chronic infection, they can treat the patient at the same time with antiviral prophylaxis. So that makes sense. I think in the future, if we have any way, to treat, with this disc RNA, similar assay can be used. A similar protocol can be used. Yeah.
Thanks.
Ling. Hi.
Dan.
That was great. So just so I understand it, the circular RNA is basically a competitive inhibitor of PKR, right? So and that's independent of upstream nucleotide sensing pathways like RIG-I or CGAS STING. Those even if they were active, you would still get inhibition of PKR with this circular RNA. Is that the model?
Yeah, so actually I skipped a large part of the, before previous study. So actually, if we think about the homeostasis of this RNA, when RNA is getting activated, it degrades everything, not only circular RNA. Also degrades cellular RNAs, cellular mRNAs as one way to antivirus, and also degrades the viral mRNA. But the difference between the linear and circular RNA is those linear, they can rapidly compensate, they have very quick transcription. But circular binding is very slow. So that gives the circular a very unique feature that after their degradation, it takes a long time to recompensate to the high level. I think that's the essential part in vivo that makes them can do something with PKR. So in terms of RIG-I, at least for in vitro synthesized circular RNAs, for example, from Harvard Chance Lab by Grace Chen, who's now a faculty at Yale University, they have shown that some catalytic intron protease circles, they can trigger RIG-I, but the mechanism still remains unknown.
Lily, so these circular RNAs from these exons, right? So they have to have certain features like a double-stranded RNA of certain length in order to inhibit PKR. Do you think it's under evolutionary pressure to conserve those structures, or are they just sort of have these random structures happen to be able to inhibit?
That's a great question. Actually, I have no answer for that. Because if we look at the circular RNA evolution, that's different from other type of RNA. If we only look at the sequence, they came from exon, so they are conserved. However, the same gene, they have different exons, have different circles that may cause some differences. So I think that worth to give more attention in the future. Thank you, Yishan.
Any other questions? Nope. All right. We got one.
Guess I held off. It seems as though when you express these in mice, there doesn't seem to be any effect. But do you know if but they also seem to be quite potent. Do you know if there are off-target effects or things that are other things that are stimulated? Mm-hmm.
This is a good question. First of all, for the mice carrying on those disc RNA overexpression, we have to do more stimulation. This is number one. Secondly, because in addition to PKR, there are several other nucleic acids receptor binding protein, ADAR, OAS, and NF19, F110. They all bind to degenerative double-stranded RNA region. So for the ones we made specific for PKR, that is for PKR. I'm thinking probably in the future, when double-stranded RNA in the circles can provide some unique platform to discriminate for all of those other nucleic acid receptors to have very specific inhibitors. That's one thing on the optimistic part. However, on the other hand, when the circular RNA, especially in cells, they have all different lengths of double-stranded RNA in the middle. So suggesting those RNAs may also interfere with other, double binding proteins. They all recognize degenerative substrates. So thank you for the question, allow me to do this clarification. Thank you.
All right, let's thank our speaker for a wonderful, impressive talk.
Thank you.